everyone, welcome to Pharma Journal. I am Gaurav Sharma. Today we will discuss about question answers of Analytical Method Validation as per ICS Q2.
First, very basic question is what is purpose of analytical method validation? The purpose of analytical method validation is to demonstrate that a specific analytical procedure is suitable for its intended use and provides reliable and accurate results. It ensures the method's performance meets predetermined acceptance criteria. Guideline reference ICH Q2. What are the key parameters evaluated during analytical method validation? The key parameters include accuracy, precision, specificity, linearity, range, robustness, and detection limit. Guideline reference USP Chapter 1225. How is accuracy assessed during method validation? Accuracy is assessed by comparing the measured values against a known reference standard or by performing recovery studies. Accuracy should be within an acceptable percentage of the true value. Guideline reference USP Chapter 1225. What is method precision and how is it evaluated during method validation? Method precision refers to the closeness of agreement between multiple measurements of the same sample under similar conditions. It is evaluated through repeatability and intermediate precision studies. Guideline reference ICH Q2. How is specificity demonstrated during analytical method validation? Specificity is demonstrated by assessing the method's ability to distinguish the analyte of interest from other components present in the sample matrix. It is commonly evaluated through interference studies. Guideline reference ICH Q2. What is linearity and how is it assessed during method validation? Linearity refers to the ability of the method to produce results that are directly proportional to the analyte concentration. It is assessed by analyzing a series of standard solutions across a specified range and evaluating the regression line. Guideline reference USP Chapter 1225. This one is very frequently asked question. How do you determine the range of analytical method? Answer is the range of an analytical method is determined by analyzing a series of samples with varying concentrations to ensure that the method is reliable and accurate over the intended concentration range. Guideline reference ICH Q2. Next question. What is significance of evaluating method robustness during validation? Method robustness assessment determines the method's capacity to remain unaffected by small deliberate variations in method parameters. It helps to ensure or establish the method's reliability under normal laboratory conditions. Guideline reference ICH Q2. What is the detection limit and how is it determined? Answer is the detection limit is the lowest concentration of an analyte that can be reliably detected but not necessarily quantified. It is typically determined based on the signal to noise ratio or by using statistical approaches. Guideline reference ICH Q2. This is one more important question. How do you validate analytical methods for stability indicating assays? Answer is stability indicating methods are validated to detect and quantify the active pharmaceutical ingredient, that is API, and degradation product over the course of drug product shelf life. Stress testing and forced degradation studies are essential components of this validation. Guideline reference ICH Q1A. Explain the concept of intermediate precision and its importance in method validation. Answer is intermediate precision evaluates the method's reproducibility across different days, analysts, instruments, etc. It demonstrates the robustness of method beyond the inherent variability. It is crucial or important to establish the method's reliability under routine laboratory conditions. Guideline reference ICH Q2. This one is very important and frequently asked question. What is difference between limit of detection, that is LOD, and limit of quantification, that is LOQ? Answer is LOD is the lowest amount of an analyte that can be reliably detected but not necessarily quantified. Yellow Q is the lowest amount that can be quantified with acceptable accuracy and precision. Yellow Q is usually lower than yellow D. Reference guideline ICH Q2. Next question, how do you handle validation of chromatographic methods such as HPLC or GC? For chromatographic methods, validation involves assessing parameters like peak resolution, tailing factor, system suitability, and column efficiency. Additionally, chromatographic purity, system suitability, and precision at various levels are also evaluated. Reference guideline USP chapter 621. What is the role of system suitability testing in analytical method validation? System suitability testing ensures that the analytical system, including equipment, chromatographic conditions, and sample preparation, is suitable for the intended analysis. It verifies the system's performance before starting the actual analysis. Reference guideline USP chapter number 621. How do you establish the appropriate sample preparation techniques during method validation? Sample preparation techniques are established by evaluating factors such as extraction efficiency, accuracy, and precision. The method should ensure the stability of analyte during the preparation process. Reference guideline ICH Q2. What are the acceptance criteria for precision during method validation? Answer is, the acceptance criteria for precision are typically expressed in percent RSD, that is percent relative standard deviation, for repeatability and intermediate precision results. The values should fall within the predefined limits based on the method's intended use. Reference guideline ICH Q2. Next question, how do you address the issue of matrix effects during method validation for complex sample matrices? Answer is, matrix effects are addressed by spiking known amount of analytes into the sample matrix and comparing the response to those of pure standards. Recovery studies can help assess and correct the matrix effects. Guideline reference ICH Q2. This is very important question. What are forced degradation studies and why are they performed during method validation? Answer is, forced degradation studies involve subjecting the analyte to various stress conditions, example heat, humidity, light, acid, base, to identify potential degradation pathways and impurities. These studies help to demonstrate the method's ability to detect and quantify degradants. Guideline reference ICH Q1A. Next question, how do you validate bioanalytical methods such as assays for pharmacokinetics?